Welcome to another episode of the Red Cell series. In this episode, we shall discuss iron physiology and its clinical relevance. The reason why we're discussing iron here is because iron deficiency anemia is the most common anemia in the world. Let's get started. There are two types of absorbable dietary iron. These are heme and non-heme iron. Heme iron is most commonly Fe2+ or ferrous iron, whereas non-heme iron is most commonly Fe3+ or ferric iron. Heme iron is derived from hemoglobin and myoglobin of animal sources, so this includes meat, poultry, and seafood. It is more easily absorbed compared to non-heme iron. Non-heme iron is most commonly derived from plants, and again, it is less easily absorbed. Now, we consume approximately 15 to 20 milligrams of iron from our diet. 10% of this is absorbed, so for every 10 milligrams we ingest, we absorb 1 milligram. The requirements of iron for growth is 0.6 milligrams per day. However, this increases in pregnancy to 1 to 2 milligrams of iron per day. The requirements for males over 18 is 8.7 milligrams per day. In women between the ages of 19 and 50 years, or premenopausal women, they require 14.8 milligrams per day, because blood and thus iron is lost during menstruation. Women over 50 or postmenopausal women require 8.7 milligrams per day, similar to males over the age of 18. Now, as I've mentioned, the most common iron that we ingest is ferric iron, Fe3+. This is insoluble. Now, regardless whether we ingest ferrous iron, the majority of that is oxidized with oxygen, forming ferric iron. So this here is very important. It shows that the most common iron that we ingest is ferric iron, and thus a mechanism has to be in place to convert ferric iron to ferrous iron so that it can be absorbed. And this is what we have here. So if we go through this, iron is most commonly absorbed in the duodenum plus minus the proximal jejunum. So some can be absorbed in the proximal jejunum, but most commonly it is absorbed in the duodenum. Now, exam questions like to ask you what is absorbed in the duodenum, the ileum, and the jejunum, or ask you where is iron, folate, B12, bile, amino acids, carbohydrates, and fatty acids absorbed. Iron is absorbed in the duodenum or can be absorbed in the jejunum, but most commonly it is in the duodenum. Folate is absorbed in both the duodenum and jejunum. Vitamin B12 is importantly absorbed in the terminal ileum, so this will be here. And bile is also absorbed in the ileum. Sorry, the folate here is meant to be written under duodenum, so folate and jejunum, not in the ileum. So this here is very important. Its clinical significance lies when you have a pathology in the different parts of the small bowel, or when part of the small bowel is resected, so this means is surgically removed. This results in the patient's inability to absorb these nutrients and might require supplementation. Now, in physiological pH, iron is oxidized to its ferric form. So, as we mentioned here, iron plus oxygen produces insoluble ferric iron. So, the oxidized ferric iron must be converted back into ferrous iron for absorption. And we have shown this here. Here, ferric iron needs to be converted back into ferrous iron. This here is absorbable, whereas this here is insoluble. So the conversion of the insoluble ferric iron to the absorbable ferrous iron is achieved by an enzyme known as ferrireductase. So this here is ferrireductase. This enzyme is also known as the duodenal cytochrome B. Now, in the presence of low gastric pH in the proximal duodenum, this enzyme, expressed on the brush border of the duodenal enterocytes, converts the insoluble ferric iron to the absorbable ferrous iron. So let's take a look at this diagrammatically. So we ingest ferric iron, Fe3+. The pH in the stomach is low because it is acidic. This pH with the iron passes into the proximal duodenum. Now here we have the enterocytes. These are the cells that line the bowel. So these are the duodenal enterocytes because this is the duodenum, the first part of the small bowel. So these are the duodenal enterocytes which line this part here and this part here. And expressed here we have ferrireductase. So ferrireductase is the enzyme that converts ferric iron into ferrous iron. And this is supported by the gastric low pH. So the low pH is what facilitates this. Now a cofactor for this is vitamin C. This is important, as we shall see later. A cofactor for the conversion of ferric iron to ferrous iron is vitamin C. This can then be absorbed into the enterocytes and dealt with, as we shall see shortly. So importantly, gastric acid plays a role in iron homeostasis, and PPI, which is a proton pump inhibitor, can reduce iron absorption. Other factors can also cause a disruption to the gastric pH, and we shall discuss these in detail in the gastrointestinal series. So what we have here is a diagram of the basic physiology of how iron is absorbed. This diagram might look slightly confusing, but we shall go through it in detail to help you understand and then recall and consolidate all the important parts. So let's go through it together. What we have here is the gut lumen. So this here will be the duodenal lumen. This here is the enterocyte. So these are the cells that line the gut lumen. We've just drawn one. Of course, there are many more, but we'll just focus on one for now. And here we have the blood. So as soon as nutrients are absorbed from the gut lumen into the enterocyte, they are transported into the blood and from there to their appropriate destination. So let's get started. Remember that we have ferric iron and ferrous iron. Now, if we for some reason consume ferrous iron, and this is usually a heme iron, so from myoglobin or hemoglobin, it doesn't need to be converted. It is absorbable. So this passes into the enterocyte via HCP1 or the heme carrier protein 1. Sorry, this is meant to read HCP1, not HPC1. Now, this carrier protein is upregulated by hypoxia and iron deficiency, and we'll talk about the anemias a little later. So ferrous iron can easily be absorbed into the enterocyte via HPC1. Now, once the ferrous iron has been brought into the enterocyte via the HCP1, it can be dealt with accordingly, as we shall see shortly, or it can immediately pass into the blood via the BRCP, or the breast cancer resistant protein, or the FLVCR, known as the feline leukemia virus receptor. Now, these two receptors aren't that important and we won't focus too much on them. They are still being investigated. But what you need to know is that we can ingest ferrous iron, which is taken up by the HCP1 transporters on the enterocytes. Now, ferric iron, as we mentioned, ferric iron is insoluble. It can't immediately be absorbed, so it needs to be converted to ferrous iron. This is achieved via the action of the vitamin C ferrireductase, which converts ferric iron into ferrous iron, and this occurs under the influence of low gastric pH. So this is very important. So within the gut lumen and in the presence of low pH from the gastric acid, ferric iron is converted to ferrous iron via vitamin C ferrireductase enzymes, which are expressed on the brush border of the enterocyte. So Fe3+ is converted into Fe2+. Now we have ferrous iron. This is absorbable. This can be taken up into the enterocyte via DMT1 or divalent metal transporter 1. Now this is very important and one of the high yield points, let's say. Divalent metal transporter 1 is expressed on the luminal surface of the enterocyte and takes up or transports the ferrous iron into the enterocyte. Now, once ferrous iron is inside the cell, it can be reconverted back into ferric iron for storage as ferritin. Ferric iron, ferritin. So ferritin is ferric iron stored. And we'll talk about the different storages of iron a little later. Or it can be immediately transported into the blood. This is achieved via a transporter known as ferroportin. You can think of ferroportin as a port that allows the ferrous iron to travel from the enterocyte to the blood. Ferro means iron and portin means port. So this is a port that allows the ferrous iron to travel to the blood. Now, once inside the blood, an enzyme known as hepcidin, not hepcidin, hepcidin, converts the ferrous iron back into the ferric iron for transport. Now, ferric iron inside the blood binds to a molecule or a carrier protein known as transferrin. So trans means transport and ferrin is the ferric ion. So transferrin binds to ferric ions. Transferrin. This is very important and also high yield. We can measure transferrin, as we'll see a little later. Now, transferrin can transfer the iron via the blood to, most commonly, the bone marrow, and this is for erythropoiesis, or the formation of red blood cells. We've spoken about this in the last part. Or it can be transferred to the liver for storage as ferritin. Now, transferrin bound to iron is taken up by the transferrin receptors in the bone marrow and liver. And we've spoken about what happens once transferrin is taken up in part 3 of the Red Cell series, so you can go back and have a listen to that. So this is a high yield, important overview of the physiology of iron. We'll speak about hepcidin shortly. The important part that you need to know is that we most commonly ingest oxidized ferric iron. This is converted to ferrous iron via ferrireductase. This is then taken up by the enterocyte via divalent metal transporter 1. Inside, it can be stored as ferritin, so it's converted back to its ferric form and stored as ferritin, or can be transported via ferroportin, via its port. When it's in the blood, it can be reconverted back into ferric iron, and in the blood, it binds to transferrin, where it is transported to the bone marrow and the liver primarily, as well as other organs. So what is hepcidin? Hepcidin is the only iron regulator, and this is very, very important. You need to understand how iron is regulated by hepcidin and what hepcidin is for you to appreciate many pathologies. Hepcidin is the only regulator that us humans have to regulate iron inside the body. It is produced by the liver, and its function, it sequesters, it locks up iron in storage sites by inhibiting ferroportin. Remember, we said we have ferroportin, which is a port that transports the ferrous iron from the enterocyte or from any other storage site. So this here can be the liver hepatocyte. So it locks up iron in storage sites by inhibiting ferroportin, thus preventing iron release into circulation. Now, hepcidin also limits the transfer of iron from macrophages within the spleen to erythroid precursors. Remember that red blood cells that are damaged or have reached the age of 120 days are taken up and destroyed by the splenic macrophages, and their hemoglobin is recycled. Can you remember what hemoglobin is made of? Remember, hemoglobin is made up of heme plus globin, and the heme part is made up of iron and protoporphyrin. So hepcidin limits the transfer of iron from the splenic macrophages to the erythroid precursors. So it limits the recycling of iron from the splenic macrophages, which take up the damaged and old erythrocytes, and recycle their components to the erythroid precursors. Importantly also, hepcidin also suppresses erythropoietin. Remember, erythropoietin is produced by the kidneys, and it is a growth factor that stimulates erythropoiesis and thus red cell production. So let us take a look at the enterocyte. This here is the enterocyte. This is ferroportin. Ferroportin is responsible for the transfer of ferrous iron from inside the enterocyte or the hepatocyte to the blood, where the iron can be transported to its destination. Hepcidin can inhibit this. Hepcidin binds ferroportin. So if we draw a hepcidin here, this here in black is hepcidin. Hepcidin binds to ferroportin. So as soon as hepcidin has bound to the ferroportin, whether this is on the enterocytes or the hepatocytes, it results in the internalization. So this here is the ferroportin internalized and subsequently degraded. So what this results in is that no more iron can pass through, and thus the iron accumulates or remains stored inside the enterocyte or the hepatocyte or whatever other storage it may be in. And this here is the only way that humans can regulate iron, by not absorbing more. If we have a lot of iron, we can't get rid of it, but we can stop taking in more. This is very important. Hepcidin can also potentiate the sloughing of the enterocytes and thus, inverted commas, excrete the iron. But we cannot excrete the iron from the storage sites such as the liver. How is hepcidin regulated? Hepcidin is regulated by a gene called the HFE gene, the human homeostatic iron regulator gene, so that's the HFE gene. Now, a mutation of the HFE gene results in a condition known as hereditary hemochromatosis, where we have an accumulation of iron, and this results due to the interruption of hepcidin synthesis. So less hepcidin is being produced. Hepcidin production can also be stimulated by inflammation, in particular IL-6. Hepcidin is an acute phase reactant, and we'll come to discuss that later. It can also be stimulated by increased serum concentration of iron. Remember, hepcidin prevents the absorption of iron from the enterocytes, as well as locks up iron in storage. So this is important. If we have a high concentration of iron, we don't want more in the serum. Bacterial infection can also cause hepcidin production, and we'll come to discuss this later, as well as oxygen. Now, these here are particularly important, especially when we discuss the different pathologies later in the red cell series. We store iron as ferritin or as hemosiderin. The total amount of body iron stores is around 600 to 1000 milligrams in an adult male. In females, it's around 200 to 300 milligrams. Ferritin is the main store. So this is the main store of iron. Ferritin is water soluble, and it is heat resistant up to 75 degrees centigrade. It is less stable than hemosiderin, but more readily available. It is primarily found intracellular, so this is inside the cells. Some can be found in the plasma, but this is in very small amounts. Now, clinically, we can measure serum ferritin to determine how much iron we have stored. Hemosiderin, on the other hand, is water insoluble, is more stable, and less readily available compared to ferritin. Hemosiderin is a hemoglobin-derived pigment that accumulates locally or systemically when there is excess iron. We can normally have some iron stored as hemosiderin, but when there is excess hemosiderin, this is usually pathological. Hemosiderin is visible microscopically, and we can use the Prussian blue stain to visualize it. And that is what we can see here in this picture. These are all hemosiderin. It can also classically be seen as brown granules, and these here are classical hemosiderin. Hemosiderin also has a higher iron to protein ratio. And these are the two main stores. Again, the main one is ferritin. Ferritin can be measured clinically. This is normal. Hemosiderin can be normal at low levels, but if these increase, then these are pathological. Now, importantly, ferritin is also an acute phase reactant, and we shall come to discuss this later in the series. So what is the clinical significance of iron physiology? We shall discuss this again and again as we talk through many pathologies, but this here is just an introductory. So let's first talk about the receptors. Ferrireductase and DMT1 are both upregulated in iron deficiency anemia. DMT1 mutations can cause microcytic anemias and liver iron overload. If the duodenal mucosa is damaged for any of these reasons, then the amount of iron that we absorb is significantly decreased. This includes duodenal ulcers, celiac disease. Remember, celiac disease characteristically affects the duodenum. Tropical sprue, which is a mimic of celiac disease. Crohn's disease, which is an inflammatory bowel disease, which can affect any part of the gastrointestinal tract, including the duodenal mucosa. Inflammation is also important. We mentioned that hepcidin is an acute phase reactant. Hepcidin as well is very important. We mentioned that if we have a mutation in hepcidin, we end up with decreased hepcidin, which results in hereditary hemochromatosis. And iron, as we shall talk about later in the red cell series, a deficiency results in a microcytic hypochromic anemia. In the next part, we shall talk about red blood cell indices and iron study investigations and introduce anemia. Please like, subscribe, and share our content with your friends and on social media pages. Our mission is to develop NeedToKnow video content and question banks that remain free for your use. We are unable to keep doing this without your support. Thank you.